Il progetto CoMPaSS-NMD cerca di rispondere a una domanda fondamentale. Quali sono le basi genetiche, quindi le variazioni all'interno della molecola del DNA che ognuno di noi porta, che possono spiegare perché e come la malattia si sviluppa? Sostanzialmente questa è la grande domanda. Questo progetto ha delle caratteristiche uniche, direi, perché vogliamo sfruttare appunto differenti gruppi di dati, quindi useremo dati che vengono dallo studio del DNA di pazienti con malattie neuromuscolari, dati che provengono dallo studio di risonanza magnetica, dati che provengono dagli studi istologici, e tutto questo dovrebbe permettere di costituire una metodologia che permetta di analizzare questi dati in modo da trovare collegamenti che sono nascosti. L'intelligenza artificiale ha questo scopo, la mente umana ha una capacità limitata di acquisire i dati e l'intelligenza artificiale cerca di aiutare il clinico a capire i vari contenuti all'interno dei nostri dataset. Ho tantissimi dati, uso uno strumento che mi permette di capirci di più. Non delego la mia capacità di discernimento, ma di sicuro se qualche cosa verrà scoperto potrà essere utile perché lo potrò aggiungere al mio bagaglio da professionista. E ovviamente speriamo di aumentare la capacità di far diagnosi.
Welcome to everybody. This is Filippo Santorelli. I'm today acting as the chairman of this second webinar on the multiple activities undertaken in CoMPaSS-NMD. Today the agenda is very tight, so we have first an introductory from Professor Rossella Tupler, who is the coordinator of the project, and then we will have a major discussion from Professor Joanna Polanks, and then the final questions or final section on questions and answers. Rossella Tupler is a professor of medical genetics at the University of Modena and Reggio Emilia, and as I said, is project coordinator of the CoMPaSS-NMD. Rossella, the stage is yours.
Thank you, Filippo. Thank you very much. This is our presentation. And what is CoMPaSS? The more the time passes, the more I can see what we thought to become true. So we wanted to use artificial intelligence in order to understand more and to provide a better diagnosis with patients with a neuromuscular disorder. This is an innovative project, and it was really the projection of our idea that were grown during several years of experience in neuromuscular disorder. We felt the frustration of not obtaining a diagnosis. At the same time, we were following the advancement of new technologies, the possibility of obtaining a lot of data, and the possibility of building something new. So the idea was to put together centers with different expertise to generate on one side robust and reliable data sets that can support the development of new tools in order to create a solution integrating all the knowledge. So in general, a clinician uses her own personal knowledge to make a diagnosis, to create a pathway to diagnose a patient. Now we want to standardize and to create a general tool that could be used by more clinicians without waiting 30 years of clinical experience. And at this point, to create these tools for the analysis, but also a repository to collect data, the Atlas NMD. And why do we want to do this? We want to do this because there is something that we don't know. It is frustrating not being able to provide a diagnosis. During the time, we have learned to use DNA, but this was not sufficient. It was clear. And so we thought we should use artificial intelligence because it gives us more strong, better tools in order to process data. And today, this is what you will hear from. And so the CoMPaSS use data that have been already collected in previous European studies, and then use all the experience in order to generate data from a cohort of 500 patients that will be analyzed using whole genome data, clinical standardized tools, and MRI and histology data, in order that will be deposited in CoMPaSS, the Atlas, and then this all will be analyzed by artificial intelligence in the center directed by Professor Polanks, and this because we want to obtain the instrument that will be used by the patients, by specialists, clinicians and other specialists, pharmaceutical companies, in order to obtain better stratification of patients for possible clinical trial. In general, the scientific community, because CoMPaSS has the potential of showing what can be done with data, and then policymakers that can create new ways to protect people, to make the health care more efficient. So the CoMPaSS-NMD Collect has clinical centers that are located in Finland, UK, France, Germany, and Italy. Then to an IT company, Fincos, a company with several expertise, Technalia, SIGAT, which is a company expert in molecular diagnosis or in DNA sequencing, and then the Institute of Professor Polanks, in which all the data will be analyzed and the computational tools and the artificial intelligence will be used, and Deep Blue, which provide the communication support. All this all together in order to get the interdisciplinary expertise and communication based on several years of experience. And this is a possibility to get more information about how to participate to the clinical study of CoMPaSS, and I want to thank everybody for paying attention to this presentation. Thank you very much.
Thank you, Rossella. Now I guess the audience has a more detailed, a full overview of what CoMPaSS-NMD is and the different strategies we will employ to disentangle this, the diagnosis of inherited muscular disorders. Now it's time to move to the topic of this webinar, artificial intelligence in neuromuscular disease diagnosis as a sort of alliance between patient diagnosis and technology. And it is my pleasure to introduce Professor Joanna Polanks, who's professor of computer science and biomedical engineers at the Silesian University of Technology in Poland. Joanna is an expert in data science with her research focusing mostly on developing and applying innovative algorithms for data analysis in medicine and biology. She likes a lot to develop new tools for machine learning, AI, for analyzing genetics, epigenetics, transcriptomics, proteomics, and imaging informatics. Joanna is also a very good friend of us, and she helps us to understand more in a very simple way without the technicalities that similar in this world. She helps us to understand how AI can assist in medical care. So, Joanna, the stage now is yours.
Thank you so very much, Filippo, for so kind introduction, and I'm really honored to be here. And good morning, good afternoon, or good evening to everybody who decided to join us today. We will focus on the artificial intelligence and how that, let's say, new technology can support NMD diagnosis and treatment. So that's the topic of my presentation. But there is a lot of discussion about artificial intelligence around, so it's very important to know what we are talking about. I think it's crucial to understand. So artificial intelligence, I think we may say the beginning of all the idea is like 1950s, that brilliant mathematician Alan Turing, he introduced the term of machine intelligence, and he said that he accepts that machine is intelligent if the machine can trick humans. And that was the very first idea. It took a couple of years till the idea spread around among the other people, and in 1956, really there was a very important meeting for all of us who work with the data. And then that was the moment when the John McCarthy from Stanford University introduced the idea of the artificial intelligence. And it means that it's a science. So originally it was understood as the science about creating the intelligent machines. In general, when you think about artificial intelligence, is like something being slightly more complicated than the regular calculations. So we may think about the computer, the standard one you are used to use is like a calculator. So you put some steps, you want 2 + 2, so you press the particular button, so you'd make a program, and it is done. When you think about the intelligent machine, it's slightly more behind that, beyond that idea. So you teach the machine, for example, like I have here on that screen, on that example, you teach the machine how the apples, how do they look like, okay? So you give some examples, you put some descriptors, and then the machine at some point is trained, so the model is built. And it's the good thing is that it can recognize not only the red apples, if they were in training data set, but also by similarity, there is a high probability that the machine will recognize also the green apples or the yellow ones by similarity of the other features. So we do not need, for the artificial intelligence or intelligent machines, we do not need to precise everything. And that's at least the beginning of the mimicking the human thinking and human understanding and human learning. But the those thinking machines are around us. And it's really, we use them in daily, let's say, routines. So for example, the AI systems are when you have that can recognize images, are those which you have the car plates and you go to the car parking slot, okay? And then the system camera, the vision system recognize your car, and then you don't need to bother about some tickets you put and to be able to get out or into the parking. But it's not only there. There is that recognized voice command systems. So these are the systems that allow you usually when you call some customer service, it's your first, let's say, thing you have. So then it can recognize your comments and even ask some very basic questions, and based on the reasoning, connect you to the people, to the person that is the best for your issue. And the recommendation systems like Netflix, Amazon, platforms like Spotify, they recommend you the new products, the new movies, the new kind of songs based on the historical of your kind of profile and what the similar users, similar to you, choose in their let's say when they listen or they watch the movies. So these are also the very intelligent machines that are let's say very popular nowadays. Also, the other example is like filtering emails on your Gmail or whatever mailbox system you use. You have those spam filter, and spam filter is the thinking machine because it adapts. Okay, so it starts with some particular profile of the spam emails, but then it goes next level and adapts to the new ones. So that's also the thinking machine. And also those more complicated tasks, we don't think about them that way. For example, weather forecast, it collects the data, historical data and the current data from thousands of sources of different type. And then you have these mathematical models that digest that information. And finally, we get something we're used to do and we start day with. So we go and see what is the weather forecast for today or in a week or in 2 months ahead. So these are those machines, but are they really, may we have some use of them in medicine? And the answer is yes. So artificial intelligence is playing a really crucial role in medicine nowadays and supports doctors and researchers and patients in various aspects of diagnosis. And I am going to tell you a little bit about that in details. So when we think about the applications of AI in medicine, there are several options we can think about or go for. So here is the summary of 10 of them. I think the most important one. So it's the disease diagnosis and drug discovery and repurposing. Of course, medical imaging analysis and personalization of the treatment plans. We may think also about those virtual health assistants. They are also part of the artificial intelligence thinking machines, but also that remote monitoring, that telemedicine in support of those people who need like 24/7 monitoring. And we also may think about precision surgery. Right now, these tools, these machines, they help surgeons to do proper and more accurate operations. Health informatics is something which is also a part of our daily life. When there is a managing behind the health system, there is a managing of a lot of the data, health records, the results of the tests. So they have to be stored and organized the proper way. But using that information, we build the systems which are the predictive systems. So they forecast the outcome of the treatment and the particular protocol for the patients. And finally, is something which is the medical research. Thanks to that, we can develop the new tools, gather new knowledge, and these are the next steps in the future of the medicine. So these were, of course, the 10th, the most important one, but I'm not saying the only one. So when we think about neuromuscular disorders, these two elements that were mentioned already by Professor Tapler is the analysis of the imaging data, so those from MRIs and from histology, from your biopsies, and AI algorithms can really support that part, and we'll get into details in a moment. And also the genetic data analysis. So these are the systems that allowed you to interpret, but also to look for the new discoveries, for the new, let's say, explanation of what is happening and what causes the disease and the development of the disease. But these are not the only steps we have. So for example, you can imagine that those automated screening tools are supporting also the analysis of the movement of those people, of the potential patients, or also, so it's the Kinect systems. So these are the kind of sensors you put on the body of the person, like these blue dots here you see on the left side, and then you ask a person to do very basic movements, or some which are very specialized and characterize particular disease, and then you may follow with the sensors the movement and then analyze how proper these movements were, or maybe there are some limitations and should be addressed, and maybe go for the more diagnosis. And that could be done with the webcams. So it's a future of the screening programs, especially those of highest risk. And also the precision medicine or personalized medicine is something which is a big element of the part of the medicine nowadays, or the future medicine. Is about looking what is the results of the diagnosis of the diagnostic tests for the particular patient, but also it looks on the historical data, on the current data from hundreds of patients already, and using also natural language processing, it can digest these, let's say, handwritten notes or the digitalized notes together with some imaging data. So all different types of data could be put together and then look for the analyze and propose the best treatment for the patient based on what happened in the previous, let's say, for the other patients. But let's focus on those algorithms for the image processing. These two, let's say, modalities were mentioned already. So one of those is muscle biopsy, and this is the very crucial and very important element in diagnosis. And for the moment, it requires a very highly specialized, let's say, expert to analyze this data and to look at what's happening in your muscle. And it takes time, and we know that everybody has to wait till the evaluation of those slides is done. But the technique itself is not the new ones. So in 1840s, Johannes Müllers proposed that idea of looking into what is happening in the tissue sample with the use of particular chemicals that allow to emphasize some elements, some structural elements, and some molecular components of the tissue sample. So starting from the 19th century, the idea of that histology, histopathology, and to support that in the treatment and the diagnosis developed, and nowadays we cannot imagine not having that techniques. In the down line, you see different options that people developed because they ask different questions, and different chemicals, different protocols allowed to look into the same tissue sample but gather different information. But in general, if we think about the development of the virtual pathologies that at least will support these experts and make that tedious work easier and faster, there are those several steps that must be undertaken. So the first one is to do the biopsy itself. So you have to collect the tissue sample, which is not the nice part, but sometimes is needed and is necessary to be done. Then you have to do the staining, all those procedures, and finally this ends up in that scanning system. So you have a microscope of very high zoom, very high quality, and every piece of the tissue is scanned, is digitalized, and these files are really the huge ones, much bigger than the photos you have in your phone, for example, and that is then analyzed by the experts. And what we are doing, we are working on the system which allows to do it at least automated way and suggests some information to the expert. And to be able to do this, to follow the steps that are done by the pathologist, first of all, you have to, from that all the scan, which is the regular image of the slide, of the window, of the glass part, you have to be able to automated way identify the tissue, then analyze the tissue in details, so go to the cell level and then describe every single cell, for example. So how it looks like. So here you have an example how the tissue fragment from the glass looks like. So first the system has to find that part in the image, and then we would like to be able to look in details into those different cells and to be able to segment them and to describe them in numerical way, because please remember, computers work on numbers. So even if you have an image or whatever data we have to somehow translate that into the numbers which are well understood by the computers. So it looks that these are those several steps we have to proceed with. So first we have to identify the tissue on the image and skip all the background and neglect that. Then you have to do some processing to be able to really identify and see these borders between the cells. So very often it calls for some binarization, for some quantization. There's a lot of operation you have to do. Then you segment, so you identify cell by cell, as these are those different colors for every single cell. And then having each of them, you go into the structure. So every cell must be described by its shape, size, and like uniformity of the color of the staining, like that. One of those is totally different from the other one. And we would like to be able to identify these and address that differences. So thanks to the tools which is named histomics, and having every of those single cells identified on the right part of the screen, you see the example how the fragment of the tissue scan looks like after that processing. Every color is a different cell, and each of that cells is then numerically described by like 100, sometimes 500, and sometimes in some situation even 1,000 numerical values. Okay, and having these numbers collected, so per each scan, per each cell, these are hundreds of thousands. So please imagine how big these numbers are, the data are. You look at the differences across the diagnosis. So here is the example of one of those features that describes the mast cell variability across the scan, okay, across that tissue region. So as you see on the very left, you have that shadowy, let's say, shape is the location of the values across the cells. So if you look at the normal cells for several or for hundreds of patients we analyzed, the cell size is the variability is pretty low one. So you see that plot on the left is pretty narrow. On the y-axis there are not so many differences. There are single cases when it changes. But then when you have those different diagnoses, so on the let's say bottom, you have the names for those diagnoses. So you see that, for example, for IBM, then you have a huge variability, much bigger variability of the cell size, which means that you have a very tiny cells and you have a very big ones, and they are not similar on the tissue over here. And the specificity differs, of course, depending on those diagnoses, but thanks to that, we may use those hundreds of numerical descriptors and then try to predict for the particular bigger system that using those characteristics, morphometric cell characteristics of the cells, will be able to classify the slide to at least suggest the diagnosis. So that's the option we are working on, and we are pretty successful. A second imaging is about the magnetic resonance. Is usually what you do when you would like to look at the structure, internal structure of the muscles, not only tiny biopsy. So these are that technology is also very frequently used in NMDs, neuromuscular disorders. And then if you would like to analyze this data, you remember these are the data look like a lot of those single slices that were collected in the acquisition of the images. And then you have to do some kind of pre-processing to identify the background and to focus on the muscles only, and so on and so on. And then you have to do the feature extraction. That's the operation that allows you again translate image into the numbers. And based on those numbers, you may perform the classification. So you may say that the muscles in the leg look similar to the other patients, or you may do the task which is named classification. So you may classify or predict based on the previously available data, the new patient to the group of similar kind of properties. So that image pre-processing step requires a lot of the mathematics and IT skills. So you start with the general image, then you have to do the segmentation of the body. You have to identify that subcutaneous tissue, and then bones, because we don't want bones to somehow be incorporated in our analysis, because muscles are of our interest. So these are those several steps. Each of those is a separate algorithm and separate tool developed. Then you have to, if you would like to make a description, you have to focus. So you can focus on particular muscle of your choice. Or you can say, I'm looking for a complete kind of a description of the tide. So you go for all the slices which are automated way identified as belonging to the tide. Or you can go for the 3D image for the body part. It's on you. But whatever choice is yours, those steps which are required, that pre-processing and skin identification, again, they were developed by us. And then we have, oh, sorry, it looks that there is no, on the very right, we lost in that PDF transformation, we lost the extra part, which is the definition of the sub-regions, which are like pies. You can imagine the tide being a pie, and you cut the pie into slices, and then we analyze these slices separate because they are related to particular muscle group of muscles. So having this done, you go for the next step. So again, you calculate those numerical descriptors as we did for the histomics. They are slightly different defined, but this is a bunch of numbers that allows you to go from image to the number numerical representation. So because it's a lot of those slices, there are different numbers of them. You have to somehow standardize and sample them, and then what you do the projection. So there are those artificial machine learning tools that allow you to go from a very high dimension, so huge numbers describing your MRI scan, into one dot. So every single dot here on that left part, if you to top left, let's focus on that, is one patient. Okay, we did all the steps I've just introduced to you, and it's one patient, is one dot. And then we color code them with depending on the diagnosis, okay? And if the dots are closer, it means this MRI image is similar, okay? It's very similar for those two patients. If the dots are separated distant, we claim they are different. And when you see, it's a nice kind of projection of those. So, for example, the green ones are the limbo-girdle muscular dystrophy type 2A. These patients, they nicely kind of locate on the bottom right part of our visualization. GNE myopathy, these blue dots are on the very left one. So as you see, some groups, they nicely cluster. We use the word cluster. Some of them not very, let's say the diseases when we do not have so many examples, that are more spread around. So if we trust that kind of representation, we can go for another, let's say, for the next step, when instead of following the diagnosis, we look at the similarity of the images, and then we identify a group of patients with the similar, let's say, imaging data from all those properties. And then we see if it's consistent with our diagnosis. It's more or less consistent when you compare these two color codes here. But I would like to emphasize one extra part, which is here. So in that CAPN3 patients with that diagnosis, on the bottom right of the left plot, you see the pinky color being all around in those two legs, if I may say, that it looks, our plot looks like a teeth. So we have those two roots for the teeth, let's say that way, the pinky ones. And our system AI diagnosis identifies two groups. And the question is why it's happening that way. And if we go into more details, you can right now see easily that these patients from the bottom part, the green one, they have totally different level of severity of the disease when you compare to those being on that, let's say, yellowish part above. These guys, the brighter the color, the more, let's say, fat and the more collagen, less muscle is there. So you see they are very different for the machine learning system, and that's why they clustered them as a separate groups of patients. And when we think about the genetics, of course, we may put more time, like another lecture about the genetics. But in general, when you have that diagnostic workflow for right now, it's the idea that you have that clinical evaluation. You evaluate the weakness of the muscles, the onset. You look at the history, family history, all those things, and then you check first the targeted, the genetic of, let's say, the genes which are candidates for you, clinically suspected ones. If it doesn't work, then you look for the gene panel. You try to fit your patients and to identify one of those. If it doesn't work, then you start investigation. So you'd go for the whole genome, whole exome sequencing, and you look what is so specific for that particular patient or group of patients, and that causes the disease. If that doesn't do the job, then you go for the copy number analysis. So those structural variants, the very advanced and sophisticated things. So the first three steps are well known and are used in clinical practice. Right now, we are focusing on that knowledge discovery for those undiagnosed patients. By undiagnosed, I understand the patients that do not fit into any of those criteria currently used. And when you ask about how can AI help in the process of that diagnosis, so first of all, together with this, let's say, investigation of the and variant interpretation, a very detailed one, also we can go for the phenotype recognition. So based on the general description collected by doctors and by, let's say, using that natural language processing tools, ChatGPT, everybody knows that. So then, but it's dedicated to that particular task, which is the phenotype recognition. We analyze the data, and then based on that, there is some candidate disease categories of genes found by AI system. We can also do genotype phenotype matching, or, which is very, very, very hot topic, I would say, in AI now, identify those therapeutic strategies based on the looking for the best match among the patients with the similar genetic test results, and then we can see what drugs or what therapy was used, and then should suggest the one for that particular patient, even if we don't know what type of disease it is. So just to summarize, if you think about the what can artificial intelligence do in case of neuromuscular disorders, we have those three areas. So it's diagnosis, it's treatment planning and monitoring of the disease. So we've mentioned already about some pattern recognition in medical imaging. I didn't mention the electromyography interpretation, but it's also a separate part. Then we have that genetic data analysis. For the treatment, we can go for that personalized or precision medicine based on the historical data and analyzing the already existing protocols and the outcomes. And then we can predict the disease progression, also building particular models dedicated to that task. Or also, it was not mentioned during my talk, but it's a limited time, optimizing the rehabilitation with the use of that particular, for example, that vision system that is able to analyze what is the weakness of the particular patient, and instead not only screening system, but also these rehabilitation, interactive rehabilitation things. And finally, the monitoring of the health, of the status health, all those variables we have around us, they can help us, and also the telemedicine and the voice and movement analysis, just to evaluate the progression of the disease. So that's the I think the short introduction to the artificial intelligence in neuromuscular disorders. But all those tools and the methods we've developed are, of course, done by the wonderful people with support. So these are my people working within that project, Michał Marczyk, Michał Markiel, and we have a support of those wonderful, great experts from the Munich and Pisa in case of the histology. And then when we think about the MRIs, these are those two wonderful young guys, very enthusiastic, working on that system for the MRI analysis with the support of our wonderful, let's say, experts from the Newcastle University and the PhD students from that center that works together with my young enthusiasts. And finally, about the genetic data, we've developed a system that allows to identify what is so specific and which signaling pathways are, let's say, affected by mutation and variants for the patients. And Alexandra and Carolina are those two young scientists working on that. And of course, we couldn't do anything with the people who are guiding us toward the great solution. So it's Professor Tapler, you've already met, Marco Savares, Anna Top, and Filippo Santorelli. And thank you so very much. And I'm very open for any questions you can ask now, or you can contact me later via email. So here are the contact data. Thank you so very much for your patience and for your attention.
Thank you, Joanna. Very nice presentation. It's like a jump into the future. So we expect a lot from results in CoMPaSS. Now we have some minutes left for a few questions. This is the question and answer section. So if you have some questions, please don't be shy. Well, we have a future. What kind of future do you anticipate for AI? Let's say in 2034, in 10 years, what we can imagine we can see for AI in the medical field?
Oh, that's a very nice question, and it's not so easy to answer, because as an AI engineer, I believe that we will be able to really do the tremendous work and support doctors in the diagnosis and all the healthcare workers. Right, I think personally, in my opinion, I think that AI will be present on daily routine and will support the doctors, but it will not substitute the doctors. That is very important, because the systems in general, they work very well for the cases, the average situation, average case, average patient, let's say that way, and it's as good as the information it collected for the training and during the process, during the usage. If there is something new, it may also suggest, but will never ever take responsibility for the diagnosis. So I think AI in medicine will be very, very present, making life easier and some procedures faster, as automation did. But the doctor will always be the person who decides at the end, and we will not, I believe, will not resign from the support of the humans like the nurses, because the personal contact is also necessary, that for the well-being of the patients.
Thank you. There are other questions? You took the tabular radiomics data from all muscle and did the clustering algorithm, right? When you extract the radiomics data from the muscle, do you segment away small blood vessel and other irregularities? This is the question for you.
Oh, that's a very nice question. Okay, I will try to answer to that. Of course, it would be very temptation to go and do all those segmentations, those small blood vessels and all other things, but we are limited to the resolution and the quality of the data collected. If everything depends on that. So if we have a high resolution scans, of course we do that. But if you have these 500 by 500 pixels, for example, then you cannot go into details. So you classified or you identified the main structures, which is the muscle, the bone, the collagen, the fat, all those things. But you cannot go to those small blood vessels at that resolution. So we do these things, but everything depends on the quality of data which are available. I hope I answered the questions.
Yes, I guess this is the right answer. The better the data, the more
Information you can
helpful
collect.
can be the information you can get, right?
Yeah, but you have to compromise, because at some point, if the information does not change significantly the final decision, and you have to compromise if you are going to get that information, because it's usually very time consuming and computational load requirement is pretty high. So we have to compromise. Let's say the improvement may not be worth that extra work. So it's usually at the next step for the diagnosis, not the preliminary one.
And also because I guess if the system is not too much democratic, you can rely on data obtained from very few centers where you have a very high quality, and you cannot use the bunch of data obtained by the real world, real patients that you can collect in multiple centers, right?
Yes, that's very true. And because of that, I think we benefit from the design of the CoMPaSS project when we really work or develop our algorithms before we are going to analyze these 500 patients. We developed our algorithm on historical data from very different centers, different scanners, different, let's say, age and staining protocols to make it as robust as possible. But I cannot promise that it will be ideal. But still, we believe that it will be supporting and useful for the professionals.
Right. Other questions? Okay, so a question for you. Can you predict improving of the diagnostic accuracy?
I would not like to go for the numbers because, if you say 20%, you have to bring some arguments. But what I can say from my experience working with that data from the CoMPaSS project already, we see that of those, as you know, about 40% of patients or even more, that depends on the center, they remain undiagnosed. By undiagnosed, I mean they do not fit into any of those current used diagnostic criteria based on, for example, on the genetic data. So we see that a lot of those patients named undiagnosed ones or classified as undiagnosed, they have a huge similarity to the patients with already known diagnosis, but the disease is caused by slightly different mutations or slightly, let's say, other genes, but in the same signaling pathway. So based on that, I believe that at least a quarter of those patients we will be able to suggest the diagnosis, but maybe more. I'm not, because I am thinking about saying about single omics for the moment. So based on histomics or based on MRIs or based on genetics. If we combine them together, I believe it might be even better.
So the number of diagnoses, and also I guess it applies to the accuracy of diagnosis, right?
Yes.
Yeah.
Because if you, I interpret accuracy by being able to give the diagnosis, okay? But of course, there are those cases when you have misdiagnosis, okay, because of something, but it is not as frequent, I guess. It happens, of course, according to what I know. I'm not a clinician, I'm not the geneticist, so I can rely on information that was provided by my colleagues. So misdiagnoses are not as frequent as those lack of diagnosis. So that's why I'm focusing in the answer about that. But of course, we can always verify the diagnosing and see at the similarity to the other groups of patients.
Okay, thank you. I have a provocative question for you, okay? A few months ago, I was trying to explain what AI is to my mother, who is a very simple person, not well educated. And then I found some difficulties, but then she said, well, it looked like when she was young and were the first way to show on TV, and television was a sort of authoritative source of information and entertainment. She was saying, well, it was on TV like a support the truth of the statement, just a way to silence any kind of skepticism or criticism. So she said, it's perhaps, do we fear the same, like it was on TV means it's true, and it was said by AI, so means that we don't have to put more reasoning and we have to trust blindly to AI. Can you comment on this?
Yeah, it's already happening, I must say, and it's something we are aware of, and there is a lot of discussion across the engineers and the clinicians, because you know, if we use a tool and the tool was like 100 cases correct, our trust increases, and we start to not checking on validate the suggestion given by tool. And it's a human kind of property. And because of that, in the systems, right now, there is a separate, let's say, branch in AI that tries to check or kind of control the usage of the tools. So, for example, if the doctor is clicking that everything is fine with all like series of tests, and there is no intervention, no kind of longer looking at the data, it says that something might be wrong. Of course, could be that the system was correct, but it starts to suggest some kind of action from the doctor. So they do not, the kind of perception is active again. So yes, that's what's happening, and that's very true. At some point, but if I may add, I don't know if we run out of time, that enthusiasm toward AI was on the top, on the peak, I would say a year or year and a half ago. Right now, because the expectation was so high, and we couldn't fulfill all those expectations because it takes ages and it takes technology and it takes data, first of all. But still, the trust is high, too high in my understanding, especially for the medicine. We have to look, but we have to verify and use and verify always, because the machine can be wrong.
So use the tool, but filter by your brain. Okay.
Yes, it's something is suspicious. Trust your instincts, especially if you are a doctor and with the experience. So yeah, exactly. So we should put that kind of, let's say, sentence on the systems.
Thank you. Are there other questions? If not, I thank you again, Joanna, for this excellent presentation. Very instructive, I guess. I myself learned a lot today.
Thank you.
Rosella is joining us just to conclude this webinar. Rosella, do you want to add some few notes before we conclude?
Yes, I think that it is very important. We have tools, but we need judgment, because also genomics was the same. When we started discovering genes responsible for disorders, everybody was on genetics. And now, look, we have to read everything again and to re-elaborate. So this is the point.
Thank you. So now we go to conclusion. So this was the second webinar. And we have next third webinar. We'll examine how to manage sensitive health data. The seminar will be on November 25th, so it's about 1 month. The presenters of the next webinar will be Dr. Michael Orbach and Dr. Ainara Garzo, both from Technalia. Rosella Tupler will serve as chairman. Michael and Ainara will discuss about a very hot topic, how health data can be secure and handled with care. European Union regulatory bodies are very sensitive to the handling of medical data, and so this is a very hot topic. So I expect to see you guys next month on the first webinar. And that said, I thank you all for participation. Thank you again, Joanna and Rosella as speakers, and thank you all for attention. Bye-bye.
Bye. Bye-bye.
Thank you. Bye-bye.
Bye.